Hi students, in the last section we have discussed about the classification of bacteria based on their mode of respiration and the mode of nutrition. So in this section we are going to study about the various reproduction, the types of reproduction in bacteria. Okay. Most bacteria, they reproduce in the simplest form of what? Asexual reproduction. They use simple forms of asexual reproduction and very rarely by the sexual reproduction. So let's see what are the different methods of this reproduction. Okay. So, the most common method of asexual reproduction, that is known as what? The binary fission. So, binary fission, it is the most common method of multiplication in bacteria. Here the bacteria which duplicates its DNA and then divides into two equal parts. Okay, so it is direct cell division. Okay, it's otherwise known as what? Mitosis. The bacterial cell, what happens is that the DNA, it is duplicated. The bacteria duplicates its genetic material and then divides into two equal parts. Okay, so that is what? Binary fission. Then in the case of sexual reproduction, here it is bacteria which exhibit a primitive mode of sexual reproduction. And there is no formation of gametes or fusion of cells. Okay, there is no formation of gametes. It's a primitive mode of what? Sexual reproduction. Bacteria exhibit a primitive mode of sexual reproduction. And there is just what? There is an exchange of genetic material. There is an exchange of genetic material. So, it's better, this process is better known as what? Genetic recombination. Okay, so rather than this sexual reproduction, a better name is what? Genetic recombination. So let's see what are these types of what? Genetic recombination in bacteria. So, the first method that we are going to discuss is what? The transformation. Okay. The first evidence of this bacterial transformation, it was reported by a British scientist. Okay. It was Frederick Griffith in 1920. During his experiments with a bacterium, Diplococcus pneumoniae, he reported this bacterial transformation. So what is bacterial transformation? It's a process, a simple process. It's a process by which the free naked DNA in the solution is mixed with, or it is incorporated into a recipient bacterial cell. So it's a process by which free naked DNA is incorporated into a recipient bacterial cell. So that is, there is in this type of transformation, there is a transfer of genetic material from one bacterium to another bacterium. So here there is what? A transfer of genetic material from one bacterium to another bacterium. Okay, and this process, it does not involve the contact of two cells in any way. There is no involvement of the contact of two cells. So there is what? There is a transformation. Okay, there is a transformation of what? The genetic material from one bacterium to another bacterium. Okay, so that is known as what? The bacterial transformation. Now let's see the next method. The second method, it is transduction. Okay. And this method, it was first reported in the Salmonella by Sinter and Lederberg in 1952. So in transduction, there is a small piece of DNA. A small piece of DNA, it is transferred from one cell to another cell by bacterial virus. Okay. So in transduction, there is what? A small piece of DNA which is transferred from one cell to another cell by bacterial virus. That is what? Bacteriophage. Okay, and it was reported in where? Salmonella by Sinder and Lederberg. Okay. Then the last method that we are going to discuss is for the conjugation. So it is the only method of genetic recombination where there is an involvement of what? Cell to cell contact involvement is there. So there is a cell to cell contact. Okay. Then it was reported in E. coli by Tatum and Lederberg in 1946. So in conjugation, the conjugation, it occurs between a male and a female. Okay. So in this type of reproduction, okay, it occurs between a male and a female. Then what is a male? In males, male means what? A donor cell, a donor bacterium. Okay, it contains a plasmid. The donor contains a plasmid that possesses a factor, that is what? Fertility factor or F plus. So the donor cells may contain a plasmid that possesses a fertility factor. So this type of cells, it is known as what? The male. It is considered as male. Then those which lack this, that is what? Females. It does not have what? The fertility factor. And this fertility factor, it contains the genes that produce the sex pyele and some other characters that is responsible for the transfer of the genes. Okay, so there is what? There is an exchange of genes taking place. In conjugation, there is what? An exchange of genes take place between the males and females. So males are what? They are having the fertility factor, and in case of females, there is no fertility factor. So there is an exchange. So and there is what? There is an involvement of what? The two cells. So there is cell to cell contact is there. Okay, in this type. So these are some of the methods of reproduction in bacteria. So now let's see the two types of bacteria. One is gram positive and the next one is what? Gram negative bacteria. So let's see the difference between these two types of bacteria. So bacteria are classified into gram positive and gram negative, and it is based on their reaction with gram stain. So this technique of gram staining, it was introduced by Hans Christian Gram in 1884. Okay, so this technique of gram staining, it was introduced by Hans Christian Gram in 1884. So by doing this experiment, we can say that whether this bacteria, whether the bacteria is gram positive or gram negative. So these are the steps involved in the gram staining. So let's just have an idea of these steps. Okay. So first we have to make a thin smear of bacterial culture. So first we have to make a thin smear of what? Bacterial culture. And it has to be air dried. Then it has to be air dried. So this is the first step. The second step, the smear. Okay, the smear is stained with alkaline solution of crystal violet. Okay, in this next step, what we are doing is that what? the smear is stained with alkaline solution of what? Crystal violet. Then at that time, what will happen? So what we can observe at that time? That time the cells take up the stain and becomes blue. Okay. Then in this next, in the third step, these cells, they are treated with 5% iodine solution. Then it has to be treated with iodine solution. At that time, the iodine fixes the dye to the cell wall. So that is the observation. Okay, the iodine fixes the dye firmly to the cell wall. Then it has to be washed with 95% alcohol. So this is the last step. Okay, it has to be washed with 95% alcohol. Then we have to observe this under this light, this under the microscope. So if you observe it under the microscope, we can say that whether it is what? It is gram positive or gram negative. So these are the steps involved in what? Gram staining. Okay, now we when we observing under the microscope, we can distinguish these two types of, we can see there are two types of what? Bacteria. Okay. So those bacteria which retain their original crystal violet stain is known as what? The gram positive bacteria. So when we are observing under the microscope, we can see that some bacteria, okay, which retain their crystal violet stain, so that they are known as what? The positive, the gram positive bacteria. And those bacteria which loses the stain and discolorize are called what? The negative, that is gram negative bacteria. And the basis of this gram staining is nothing but the lipid content of the bacteria. The basis of gram staining is what? The lipid content of what? The bacteria. So in the case of the gram negative bacteria, which have very high lipid content, when we compare to the gram positive bacteria, the gram negative bacteria may have high lipid content, due to which the lipids dissolve in alcohol or that organic solvents. Okay, the lipids dissolve in alcohol. So along with it, along with the lipid, it will take out the stain also. Along with the lipid, the stain also taking out. Okay, and that way it will become what? Discolorize. So when it becomes discolorized, it is known as what? The gram negative bacteria. Okay, so this is the procedure involved. Now let's see what are other difference between this gram positive and gram negative bacteria. Now let's see the differences between the gram positive and the gram negative bacteria. Okay. So in the case of gram positive bacteria, they retain the gram stain. Okay. But in this side, what? Gram negative bacteria, they does not retain gram stain. Okay. Then they have a rigid cell wall. Due to the presence of the peptidoglycans, they have a high amount of peptidoglycan. There is what? They have a rigid cell wall. Here, they have more elastic cell wall. In the case of gram positive bacteria, their lipid content, it is very low. But in gram negative bacteria, they have a high lipid content. Then their cell wall, in the case of gram positive bacteria, their cell wall, it is sensitive to antibiotics like penicillin. In the case of gram negative bacteria, they are insensitive. Then phospholipids are absent in gram positive bacteria, but it is present in the gram negative bacteria. The gram positive bacteria, they produce exotoxins, and gram negative bacteria, they produce endotoxins. Okay, so these are some of the differences between the gram positive and the gram negative bacteria. So here gram positive bacteria, there is what? Streptomyces and Clostridia. These are what? Gram positive bacteria examples. Then E. coli, Salmonella, Rhizobium, then Nostoc, all these are what? Gram negative bacteria. Okay, so today we are going to conclude this. Okay, now this section is going to conclude. So you may go through your textbook. Okay, so if you have any doubt, you can message or call me. So let's meet in the next class. Okay, bye bye.